So now we want to take that green fragment that we've generated from the genomic piece of DNA, and we want to actually place it into our green digested plasmid PPOP. And so you can see here's our plasmid that's been digested with green, and then this is our piece of genomic DNA that's been digested with green. And you can see the green are going to have compatible ends so that they're going to be able to join effectively. And that's going to be the case with many restriction enzymes as well. If you have two different pieces of DNA that you've digested with the same restriction enzyme, they are going to have what is called compatible ends so that you can make ligation pretty straightforward. So you're going to ligate these by just putting them together, and then you're going to try and draw a new map with the genomic insert. Again, you want to include all the labels, things like restriction sites, the genes, and any sizes that you know, and you want to write these either on the map itself or in some sort of chart form. I actually always prefer to write it on the map itself. And then you also want to include some sort of total size of the new plasmid that's now a recombinant. So your recombinant plasmid with your ARO-A gene is probably going to look a little bit like this. Again, highly recommending that you draw this out yourself. Even though I'm basically giving you the answer, you are missing out on some valuable understanding by not drawing it on your own. So this is, we're just going to call it recombinant R-PPOP. And the new size is, remember, our insert with the ARO-A gene was 2,000 base pairs. And our original plasmid was 3,000 base pairs. And so our new recombinant plasmid is going to be 5,000 base pairs, because you're basically just going to add these two things up. It's also helpful, as you can see that I've done here, where I've actually put in how much DNA, how many base pairs there are between like this green and this yellow site, and this yellow and this other yellow site. And now we know from the plasmid itself, from the yellow to the green, on the plasmid itself is where we've got the green. This is the original plasmid here, and this is our insert region. The LAC-Z is now not going to be functional because we've actually popped an insert into that green restriction site. And so by doing that, we have now put this huge space in between these two pieces of the LAC-Z gene, which are going to render it non-functional. Again, even though this is the plasmid that we've been playing with, I am continuing to put in the base pair sizes for any of the fragments that I know, and this just allows me easier access to it so that I can kind of decipher positives from negatives when I'm trying to come up with a way to look at that. This will give me as much information as I need in order to do that successfully. So we already just briefly addressed that the LAC-Z, because it's now split, is not going to be functional on the recombinant plasmid. We've now disrupted that entire LAC-Z gene by adding an insert, in this case the ARO-A gene, and that huge piece, 2,000 base pairs, is going to render our final LAC-Z protein product non-functional, so it's not going to produce a blue colony in any bacteria that pick it up. That means it will be producing a white colony instead of blue in colonies that take up a plasmid that contain an insert. Then on the lab, it's also asking you, will the ampicillin resistance gene be functional in the recombinant plasmid? And the answer to that is absolutely. That ampicillin gene is part of our screening mechanism, so we have not done anything that is going to disrupt that gene. So the ampicillin gene better be functional because we're using that as kind of a negative slash positive screening mechanism. So we want that ampicillin gene to remain functional. It hasn't been damaged by any kind of restriction enzymes, and so it should be functional. So the new base pair size of the recombinant plasmid, again, is that original 3 kilobases of the PPOP original plasmid plus the 2 kilobases of the chromosomal fragment that we're hoping to clone in that contains that ARO-A gene from in between those green restriction sites. So again, just like with the plasmid, you're just going to add these two up and it's going to give you the new size of that recombinant plasmid. This is really important because if you haven't done, your adding up of pieces that you're adding in, you're going to end up with some very confusing restriction evaluation when you go to the next step. So it's important that you kind of consider these things as you're moving forward and that you label everything as appropriately as you can. So now we want to take our recombinant plasmid map that we've just drawn, and hopefully you have drawn it as well, and we want to use that recombinant plasmid map to kind of estimate sizes by doing different restriction enzyme testing. And so we can cut with one enzyme, we can cut with multiple enzymes. So if you see, on one of the lab parts, I guess it's yellow. If you see this and pink, that just means you've cut it with both of these. And so if you have either one of these sites there, anytime there's a yellow site or a pink site, it's going to cut. If you just cut with a single restriction enzyme, only that single restriction enzyme is going to digest. So I just kind of want to make sure, because this is, I really think, one of the most important parts is being able to evaluate your new plasmid map and being able to estimate or basically predict what the results are going to be from a particular digestion. And so I think you can do this, but I'm going to help you with the first two, and then you can kind of take it from there. So if you cut with pink on your new recombinant plasmid here, what fragments are you going to end up with? Well, if you just kind of look around across the plasmid, here's a yellow site, here's a yellow site, here's a green site, here's a yellow site, here's a pink site, here's a green site. So there's really only the one pink site. So just like when we were linearizing our plasmid from our beads, there's only one restriction site here of the pink. And so when you cut here, the only thing that's going to happen is it's going to open up that plasmid into a linear piece of DNA. So you're just going to have one piece of DNA that's going to be the approximate size of the new plasmid, which, remember, in this case is 5,000 base pairs. Hopefully that makes sense. But what if you cut with the green restriction enzyme? So if we look across, again, we've got our yellow, we're not cutting with that. But if we're going to cut with our green restriction enzyme, there's one site here and there's one site over here on the other side. And so if you have two restriction sites, it's going to cut in both places. And if you allow it to complete its restriction digest, it should be a complete digestion. So what I mean by that is it's going to basically, in this case, because we used green to clone into the plasmid, it's basically going to just pop that insert right back out. So from one green to the other green, you're going to have 2,000 base pairs. And then the rest of the plasmid, or the original PPOP, was going to be 3,000 base pairs. So in this case, because you have two restriction sites on your plasmid, you are going to end up with two separate fragments that will run out on a gel. So what I want you guys to do on this, last page of the pop bead is you're actually going to go in here and you're going to draw in where you think those fragments are going to be. So in the case of lane 2 was the pink restriction enzyme, and that just linearized the new plasmid, which remember was 5,000 base pairs. You're just going to go in and draw in a band where you think it's going to be. For the green restriction digest, we just talked about how you're going to have your 2 fragments that are going to be 3,000 and 2,000 base pairs in size, and you're just going to draw those on. And now you're going to go through and you're going to do the rest of the restriction mapping, and you're going to estimate what those fragment sizes are going to be and draw them on the gel. And that's going to be one of the pages that you're going to turn into me. So I want you to finish the remainder of the restriction analysis. And here's just, I want you to take your time on this. Don't rush it. Make sure that you are hitting all the sites, that you're adding everything you should. And so just take your time and really finish the rest of that restriction analysis. This is going to make the rest of the semester so much easier if you have taken your time and really worked through this pop bead exercise. It's really going to make a difference in your understanding of cloning. And so then turn in to me, if you would, a hand-drawn and complete map of the recombinant P-pop plasmid. I know I've provided this to you, but I cannot stress enough how important it is to draw it yourself, because it really helps to solidify the different concepts. And then I want you to complete that last page of the pop bead lab, estimating the fragments, and turn that into me. So those are 2 things for lab 6 that you'll be turning in. There is no notebook recording for this particular lab, but hopefully it will help the rest of the semester go a little more smoothly. As always, if you have any questions, comments, suggestions, issues, please do let me know, and I will see you in class.